These vintage botanical illustrations show how early botanists categorized summer flowers. To determine a plant's relatives, they studied visible traits like the number of petals or the arrangement of leaves. This diagram shows the primary divisions used to sort flowering plants, monocots and dicots. The classification relied on physical contrasts, like parallel versus branching leaf veins, and the number of seed leaves inside a sprout. However, physical traits are misleading. Through convergent evolution, unrelated plants develop identical structures to survive similar conditions, making appearance a poor predictor of heritage. Grouping plants by these shared looks meant the established classification system was often inaccurate. It documented how plants adapted to their surroundings, but failed to show their actual evolutionary paths. DNA sequencing provided a way to look past these surface-level similarities. In the 1980s and 1990s, scientists gained the ability to read molecular markers, the genetic code that remains consistent regardless of a plant's environment. In 1993, researchers published a study comparing the rbcL photosynthesis gene across 5,000 different flowering plants. This was a collaborative effort to map genetic similarities at a scale never before attempted. The resulting data showed that the traditional dicot category was genetically invalid. As this diagram illustrates, these plants didn't descend from a single common ancestor. They were actually a collection of separate, unrelated lineages. Centuries of botanical organization were suddenly obsolete. The discovery left the field without a unified structure, requiring a total reconstruction of the flowering plant family tree. Botanists began moving away from simple taxonomy, or naming by appearance, towards systematics. The goal was now to map the true evolutionary history of every species based on shared genetic ancestry. This shift changed the role of physical herbaria. These archives became genetic libraries, housing the DNA evidence needed to verify plant relationships. Technological advances allowed researchers to extract DNA from these historical samples without destroying them. Sequencing this genetic material let them confirm or correct where each plant actually sat on the tree of life. The sheer volume of new genomic data was too complex for any single scientist to manage. Organizing the entire plant kingdom now required a level of global cooperation that didn't exist before. Isolated studies gave way to a new model of science. Mapping the relationships of hundreds of thousands of species required a unified international consensus. In 1998, a collaborative group of systematic botanists formed the Angiosperm Phylogeny Group, or APG. They worked as a single organization to establish a data-driven classification standard for flowering plants. The APG applied a strict new rule. Every recognized group must be monophyletic, including one common ancestor and all its branching descendants. Groups failing this genetic requirement were dismantled. The invalid dicots were broken apart, and verified branches reorganized into new clades, like eudicots. By grounding the system in monophyletic groups, the APG replaced subjective botanical opinions with a structure built on verifiable genetic data. Between 1998 and 2016, the APG revised its tree multiple times using deeper genetic evidence. These updates often contradicted physical intuition. For instance, agave and asparagus were merged into the same family after DNA proved they were closely related. This is the palm house at the Royal Botanic Gardens, Kew. Because institutions like Kew adopted the APG system, they had to physically reorganize millions of specimens within their massive collections to reflect these new genetic groupings. The transition from visual taxonomy to genetic systematics shows how science adapts to more precise evidence. By prioritizing DNA over physical appearance, we have built an accurate, living map of how the plant kingdom evolved.